Sexual reproduction involves formation of gametes and their fusion, and hence we need to know the structure of these gametes. In this video, I am going to tell you the structure of ovum. Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure that you have subscribed my channel. So today we are back with the another video which is talking about the structure of the ovum. So if you remember, we have already done a video on the oogenesis where I have talked about all the stages of the ovarian follicle development from the primary follicle till the corpus luteum. So in that video there was one thing which is missing, is that when at the time of the ovulation, when the secondary oocyte along with some surrounding structures are released in the peritoneal cavity, what is the structure of that? Okay, so that is the ovum, or basically the secondary oocytes surrounded by the other structures, is actually going to be picked by the fallopian tube, and that structure is what is undergoing the process of the fertilization, and that is what is fusing with the sperm. So therefore it is important to talk about the structure of the ovum before dealing with the topic of fertilization. So we have already did the video of oogenesis, spermatogenesis, the structure of the sperm as well. So if you haven't watched those videos, watch it and then you can watch this video as well. So as I said, that after ovulation, the secondary oocyte with surrounding structures is going to be captured by the fibri of the fallopian tube, and at this time the secondary oocyte is actually arrested at the metaphase 2 of the meiosis 2. So this is showing this is basically a diagram of a human ovum and the surrounding structures. So we will understand first what are the surrounding structures, and then we will understand the structure of the secondary oocyte as well. All these things will actually help us the process of the fertilization. There are many events which are happening during the fertilization. Once we know what are the parts of the sperm, once we know what are the parts of the ovum, we can relate these things and can understand fertilization very easily. So here we know that the secondary oocyte is this, and this is the cell membrane of the secondary oocyte, and it is having a nucleus which is haploid, and outside it we are having the structure which is called as the zona placidosa. So this is the structure which is present, and this is the layer. If you remember, we have already talked about this in the oogenesis video. This is basically a layer which is released by the oocyte itself, and this is actually a non-cellular layer which is a proteinaceous layer. So this layer is having the glycoproteins, and this layer is having a very important function of species specific fertilization. That is, this layer is going to recognize the sperm of the same species, and therefore the sperm of a human is going to fertilize the ovum of the human. So the fertilization is species specific, and that function is done by the zona placidosa. Then after that, outside it there are other surrounding structures which are called as the cumulus euphorus. So these are basically the granulosa cells which have came with the oocyte when it is expelled outside the ovary. So there are many layers of it, and all these layers are collectively called as a cumulus euphorus, and the innermost layer is called as the corona radiata. So cumulatively all the layers are called as a cumulus or cumulus euphorus. So you can see here also that there is a large ovum which you can see, which is having lot of cytoplasm, a cell membrane. I will show you with the help of a pointer. So this is the ovum, a large cytoplasm. It must be having the nucleus as well. This is the cell membrane, and you can see here this is the first polar body. So I forget to draw the polar body here. So between the zona placidosa and the cell membrane of the oocyte, there is a structure which is called as the first polar body, which is a result of the first meiotic division. So you can see that at the end of the first meiotic division there are two cells which are produced. One is the first polar body, another one is the secondary oocyte, and you can see that how unequal division of the cytoplasm has been there between these two cells. So ovum is having, or oocyte is having, lot of cytoplasm, whereas first polar body is having very minimal cytoplasm. So then again I will show with the help of a laser pointer. So this is a zona placidosa, and outside is the cumulus ooporus. So another important point is that these cells, this cumulus cells, they are having the adhesion molecules, and these adhesion molecules are going to interact with the fallopian tube, fibry part of the fallopian tube, and thus they are going to be picked by. So this adhesion molecules are having the role which is making it able to picked up, which is making the oocyte to be picked up by the fallopian tube. And these cells, these granulosa cells, they are actually adhered to each other by the help of a protein, or basically a matrix, which is called as the hyaluronic acid. So all these points you have to be remember, because again I said, because this knowledge is required to understand the fertilization. For example, hyaluronic acid, the sperm has to reach here. It has to fuse with the cell membrane. So before that, it has to penetrate all these layers which are surrounding the secondary oocyte. So it has to penetrate through this, and for that, if you remember the video of a sperm structure we have seen, we have talked about the acrosome. So acrosome is having the enzyme which is doing the proteolytic digestion of this hyaluronic acid, and because of that it is actually penetrating. So if you don't know where is the enzyme, what is the acrosome, and what is the hyaluronic acid, what is the cumulus, what is granulosa cells, you won't be able to understand that step. Okay, so these are just the things which I have already told you. Now we will talk about the structure of a secondary oocyte. So these are the structures which are surrounding the oocyte, and then there is a secondary oocyte. So we will draw and understand this thing. So this is the secondary oocyte, which is having a cell membrane. Cell membrane is having the specific ion channels, and in the center, or not in the center, we can say, we are drawing just for the depiction that is in the center. This is the haploid nucleus. Now the cytoplasm of the secondary oocyte is actually divided into two parts. The outer part is called as the cortex, or the ectoplasm. This outer part, okay, which is a thin layer, is having cortical granules. So cortical granules are derivatives of the Golgi bodies, which are having the proteolytic enzymes, and this is very important in polyspermy, in the prevention of polyspermy. So this is also we are going to talk when we are talking about the fertilization. So these are the cortical granules, and they are also having the actin filaments. So actin filaments are also going to polymerize, and they are also going to help in the fertilization process. So again, the knowledge of these things is important to understand the fertilization. So they are having cortical granules and actin filaments in the outer part, cortex or the ectoplasm. The inner part is the cytoplasm, is the ectoplasm, which is having haploid nucleus, mRNAs, nutritive proteins, tRNA, transcription factors, mitochondria. All these structures are present on the inner cytoplasm. So you can see here that the cytoplasm, lot of cytoplasm is being there, and then it is divided into the two parts. So this is the structure of the secondary oocyte. So very important point to remember is that the nutritive proteins and mRNA and tRNA, you can see in the ribosomes. So a protein synthesizing machinery is present in the oocyte, and this is helping the oocyte to be ready for synthesizing the proteins post fertilization, which are required for the growth and the development of the zygote. And it is also having the nutritive proteins. So basically what is important to remember is that the sperm is undergoing a process where it is losing all its cytoplasm, it is undergoing the process of cytodifferentiation. The process is called as spermiogenesis, and during that process it is losing its cytoplasm. But here the oocyte is having all the cytoplasm, which is containing all the things which is required for the growth and development after fertilization. And this is, as I said, that if you see the first polar body and the secondary oocyte, the cytoplasm is lot in the ovum and very less in the first polar body because of the unequal division of cytoplasm during the meiosis. So these are the important points which we should remember. And we have reached the end of this video. This is a reference book for this is a very good book for studying, understanding fertilization and the